Our last topic for today is lymphoma. I can give us a little bit of a historical perspective of immunotherapy and lymphoma. In particular, as I mentioned before, there was some excitement. The old days, the problem was that antibodies were not very effective because they were foreign proteins. They were usually either rodent. I remember the old days with looking at Lymphoma or Campath. They were made out of rat cells and murine cells. And it's really amazing how we've come a long way when biotechnology catches up to, with respect to their developments. It's amazing. And then Rituximab came on the scene. CD20, I remember at the time when I was involved with IDEC, they were looking at anti-idiotype antibodies, and there was actually going to be, it was an oxymoron, shared anti-idiotype. Idiotype is supposed to be very specific to the unique tumor, but they were actually going to get a panel of about 7 or 8 antibodies and mass produce it, and then looking at the patient's idiotype, seeing if there was a shared idiotype, pulling it off the shelf and using it. Then they decided it's enough. It's so hard to make one monoclonal antibody with all the purification issues and all the other issues with respect to safety testing. To make 8 of them didn't make much sense. So lo and behold, they picked CD20. They used a chimeric, half mouse, half human type of backbone structure for the FC, and that really made the major difference in I would call it the springboard with respect to the treatment of mature B cell malignancies. Now, with respect to that, at the beginning, the issue was, should we use combination therapy? And the first trial that actually I published was the combination of Rituxan and CHOP in indolent lymphoma, and it showed that we had better quality, deeper responses, higher CR rates, and then soon afterwards, Kofi looked at the elderly 60 to 80-year age group with large cell and found that, yes, there was a big difference in survival. So it's been very exciting, but we've come a long way, and we haven't really, we were at the very beginning. It was the tip of the iceberg. With respect to that, we mentioned a little bit earlier, obinutuzumab, ofatumumab, and yttrium-90 labeled CD20, ibrutinib, Tiuxetin. Basically, where we're at now with respect to lymphoma is that which of these antibodies are going to be better? Are they the same? I think there's a lot of data we're going to be seeing more with respect to obinutuzumab compared to rituximab. I think trial design has a lot to do with when we look at combinations. I think that the problem that I have is sometimes when we just examine, for example, use a chemo alone and use a chemo with an antibody, 2 drugs are better than 1. I think it's self-serving that we're going to find that combination there, but we really have to do the hard trials where combine the same chemo with an antibody against the same target or a different target against an antibody and the same chemo. I don't see a lot of that. Those are going to be the phase 3 trials. I hope they're coming. A lot of real excitement actually came in, for example, Hodgkin lymphoma and anaplastic large cell lymphoma, T cell lymphoma. They both have CD30 as their major target, and Rituximab vedotin came on the scene over a year ago, and wow, what a game changer. And keeping in mind that we often have younger individuals that have Hodgkin lymphoma, and we kind of look at it in Hodgkin lymphoma with upfront ABVD, we had approximately an 80% cure rate, 70-80% cure rate. Unfortunately, is that very tough when you have the remaining 20% of those patients that are not cured with upfront therapy. And then the worst ones are that you might salvage 40-50% with an autologous transplant. Patients relapse after transplant. And this has been so exciting with Rituximab vedotin. Now, what's interesting there is with and it was called SGN35, is that it's now being looked at maybe being used earlier on upfront, maybe as a bridge to get patients that are not responding, that are drug resistant, to get them to potentially autologous transplant, or even in younger patients, we've looked at allogeneic stem cell transplant as potentially beneficial in those patients. So the FDA granted priority review as a post-transplant consolidation also in high-risk patients. This was actually the Athera trial presented in 2015 at AACR. And what was interesting there is high-risk patients, even after autologous transplant, still have a significant high relapse rate. So the idea is that when you randomize patients to get basically brentuximab vedotin versus either placebo or nothing, those patients getting the brentuximab vedotin definitely had a clear signal they were benefiting from it. So maybe what it really does is after the transplant, what would make sense to me is there's minimal residual disease, more difficult to measure in Hodgkin lymphoma with the Reed-Sternberg cell, et cetera. And if a lot of stuff, we have the ability of using PET scans and CTs, but have about a 3 millimeters. If it's less than that or single cells, we're not going to catch it. Is that we must be clearing up minimal residual disease post-auto transplant. I think that's another theme, is that trying to treat minimal. I don't think that's where we're going to end up. I think combining these therapies up front. But I think there's an issue we didn't bring up at all before, but I think it has to be brought up now. I've done some large meetings in Europe, and there was a question is, what is the cost of the immunotherapy in the context of, safety, efficacy, and low cost of ABVD? So would we take out a drug such as bleomycin and replace it with a drug like Rituximab vedotin and say, well, you know what? We have less toxicity, perhaps the same or maybe a little bit better, not 100% cure rates, but at what cost? And then there's a lot of places around the world. And it's interesting when I was in the European perspective is, it's great that it works, but why don't we just use ABVD and save the Rituximab vedotin for later? Do you have any comments on that? If I ask the panel, open up, maybe Dan first.
So the new technologies are expensive. They're very effective. Blinatumomab is probably one of the most expensive drugs, but it's something that we don't have something like that. The CAR T cells will be difficult. These are genetically engineered. The companies are, there's 3 companies, maybe 4 that are making them. Right. So I don't know. We are moving forward, and eventually these will have to come in. It's a general, national, international healthcare. How do you provide medical care when the effective treatments become extremely more expensive, in particularly in countries, and I hope in this country, when the medical service is nationalized, when the society has to pay for it. And that becomes an issue of priorities, especially in Europe, where it is a national health service. So the countries themselves have to decide, well, are we going to take this expensive treatment, or maybe it's not so, and medicine is never going to be equal. So those who will be able to afford it will pay. So this is a huge medical, social, or even political problem. And I think that's where the discussion has to be broadened.
So, but the issue also would be whether or not, if you're not getting higher cure rates, is it worth the expense for maybe less toxicity? Madhav, what do you think about that?
So you're right. I think this is a big issue in the context of whether we can afford some of these immunotherapies. But I think the one question is going to be real data in terms of figuring out one big difference between immunotherapy in principle versus many other therapies that require so-called maintenance long-term therapy is that in principle, an immunotherapy should be able to be given once or twice and then perhaps not required over long term. So if you actually do get your bang for your buck over, say, 5-year period of time, then you might actually have cost savings as opposed to a drug that needs to be given for 5 years. Those kind of economical analyses need to be done, but we actually need data to know exactly what kind of a tail of the curve, if you will, are we actually getting.
In terms of, I think the factor that most affects cost effectiveness is the cure rate. I mean, if you cure somebody, you don't have multiple treatments. And is that?
But some of these diseases I don't think will maybe ever cure. So the question is, can you take a disease such as large cell lymphoma or ALL and make it a chronic illness with rotating some of these agents? I think that's a big question.
And I think there's another. I mean, I think the therapeutic index obviously is key. Yes. And clearly, if we had a therapy that was curing, then it becomes a no-brainer, I think. But I think the other aspect of that is the cost-benefit ratio. And I think the question is, what are you defining as cost? And it's not just, in my opinion, the administration of the drug to the patient, but it's what the cost of the patient is to society. So if you have an expensive therapy that is allowing a person to continue to work, for an additional 5 to 10 years and to generate income and to pay taxes and everything else, that cost is a lot lower to society than the cost of a therapy that may be cheaper, but is keeping a person bedridden at home.
But throw this out at you, though. So we are, actually, it's funny, I saw Stu Lichtman, who I knew from training. He's the president-elect for the geriatric oncology, and his point is that we're all geriatric oncologists, whether we could believe it or not. So the issue is, I understand what you say when you have a young person working. Well, does that mean that what do we do with the elderly patients who get most of these bad diseases? A lot of them do. Does that mean that they're not productive society? We're going to send them out to the elephant dive.
You know, I think these are going to ultimately end up being political decisions.
I think you're right.
Or insurance company-based decisions, and
that worries me. That worries me.
And these are important questions. I mean, and it's good that we're having this conversation because we have therapies that that are showing some efficacy, again at a cost, and right now we're still in the infancy of what immunotherapy is going to be doing, and so I think the main goal of therapy is to show biological and clinical efficacy. But once we've shown that, and I think in the next few years we will get to that point, then we're going to deal with the real problem, which is how can society afford to pay for this?
In the last 500 years, the survival of humans has doubled.
That's true. Older patients.
Now we have doubled the survival.
Yes.
So now we're getting all these diseases as a consequence of and the question is, it's a political, social, moral issue. Who are the patients to be treated? And it's very hard for physicians because a physician wants to do the best for his own patient, and he will use anything or recommend anything. But he's also a member of society, and a physician has to think, well, I'm treating 1 leukemia patients, but then there's no money for 1,000 diabetes patients. So this is where a physician is caught in this dilemma.
I'm going to say, we could spend 5 hours on this or 2 days on it, but one last comment, Madhav?
No, I think, I mean, there are other examples in medicine, like the hepatitis C experience in the context of trying to prevent the cost of cirrhosis and liver transplantation. But the issue comes down to the same thing, which is, does a drug need to be given over a long period of time versus over a short period of time and longer bugs for it? And immunotherapy actually does have the chance, in principle at least.
Or what actually disturbs me is to optimize the immunotherapy. If we're working with monoclonal antibodies, for example, let's try to upregulate the NK cells, get a more aggressive attack, and then back off. So even though it might mean we have to use two treatments, it's still, in my mind, more optimal than using just the antibody, depleting the effector mechanisms, and just keep giving it. That doesn't make any sense to me.
But I think to Madhav's point, and we may be talking about this, but the checkpoint inhibitors really have shown that point, and that is, you look at the data from non-small cell lung cancer and melanoma, and I think now even in Hodgkin's lymphoma, with PD-1 blockade, patients are getting they had been getting a finite amount of therapy, and they have very durable responses.
No, I think that'll be the next point. The PD-1 and PD-L1, mostly the PD-1 has been investigated. Nivolumab.